Hey, this video is going to review material from Chapter 18 from Bauman, Immune Dysfunction, or as I like to refer to it, what happens when things don't work out as planned with the immune system. The immune dysfunctions covered in Chapter 18 are going to be a small subset of the material from that chapter. As we watch this video, we want to make sure to review the learning goals associated with the 4 hypersensitivities that will be presented here. But the other material from Chapter 18 we won't be responsible for. The type of different hypersensitivities can be broken down into 4 classes, which are an interaction between the immune system and an antigen, which results in the production of disease. The antigens may or may not involve a pathogen being present, and these hypersensitivities will very nicely illustrate how the immune system can react inappropriately sometimes, resulting in disease, even without the presence of a pathogen in the human body. All of these different hypersensitivities involve the immune system and an antigen, which results in some sort of disease state. We'll talk about the specifics of each of these 4 types of hypersensitivities and look at what happens in the body as they occur. The first one, the type 1 hypersensitivity, is also going to be known as an immediate hypersensitivity. We can see that there is an antigen present up here. An antigen comes into contact with cells of the immune system. This results in a series of messengers which trigger the activation of B cells and the production of an antibody. The antibody then triggers further immune responses, primarily on different cells involved with the inflammation process. We've seen inflammation before. We've seen the role of the basophil in triggering inflammation, but now we're looking at one of the actual messengers that triggers that, and it's the antibody IgE. IgE is a very minor antibody in normal circulating serum, but it has a very profound effect in triggering inflammation. These hypersensitivities come on extremely rapidly due to the presence of an antigen, and inflammation is a very rapid response that occurs as a result. So the players from the type 1 immediate hypersensitivity are an antigen and an antibody, but the antibody in this case is the IgE subclass. The IgE antibody, which is produced by B cells circulating in the blood, binds to, in the presence of antigen, to cells that trigger inflammation, the mast cells, the basophils, the eosinophils. These ones cause the very profound localized effect. Now, the antigen that can be present can be completely in the absence of any microorganism. Just the antigen itself coming into the body can trigger this response. And in this situation, if there's no pathogen present, the inflammatory response may actually be detrimental to the health of the individual. We refer to these conditions as allergies, and you can see this sort of demonstrated right here. What's going on with this patient is that they've been to an allergist who is trying to identify whether or not the patient is allergic to various compounds. In this slide, we can see these marks with a pen, and at the tips of each of these marks, the allergist has put in a small amount of an antigen under the skin. If the patient has circulating IgE antibodies which are able to recognize and bind to that antigen which has been put under the skin, this results in the movement of inflammatory cells like basophils under the skin, the release of inflammatory mediators like histamine, causing a localized inflammatory response. And so the way that we would interpret the result that we see in this picture is that the patient is reacting against the antigen at this site, the antigen here, the antigen here, the antigen here, and the antigen here, but is unreactive to the antigen at this site. Therefore, the patient is not actually producing any IgE antibodies that react with the antigen inserted under the skin right here. By using this approach, a physician can identify what antigens the patient is making an inappropriate allergic response to. At this point, there's a couple of alternatives for making the patient better. The patient can be treated with gradually increasing amounts of the antigen that they appear to be reacting against. This can induce a phenomenon called tolerance, and the patient, the alert with the allergy, eventually becomes used to the presence of the allergen. This sometimes works, sometimes doesn't work. Usually what's more highly recommended by a physician with a patient who's got a demonstrated allergy, like in this slide, is that they avoid the presence of the allergens. Avoidance means that they won't come into contact with the allergen and no allergic response should occur. The second type of hypersensitivity, type 2 hypersensitivity, is sometimes referred to as a cytotoxic hypersensitivity. It also involves an antibody and involves an antigen, but in this case the antibody is of the IgG subtype. Antibody reacting with antigen can recruit complement and activate complement, and when complement becomes activated, binding to the surface of cells, this results in cellular lysis or cytotoxicity. We can see this demonstrated here in this picture, where donated red blood cells with a antigen that's not a self-antigen on them enter into the circulation through the process of blood donation or transfusion. An antibody which reacts with the antigen on the surface of the donated blood cells binds to the surface of the blood cells, complement is activated, and then cellular destruction because of formation of the membrane attack complex results in lysis of the donated red blood cells. This type 2 cytotoxic hypersensitivity is a very rapid response, just like the type 1 hypersensitivity that involves IgE plus antigen. The IgG plus antigen plus complement reaction is also extremely rapid, and the cellular lysis that we see down here in step 3 of this process occurs within a matter of minutes after the addition of donated red blood cells, provided the patient has developed an antibody against those red blood cells. Now, we can see here that in this type 2 hypersensitivity, the way to avoid it is simply to make sure that we do not receive, if we're receiving medical care, blood that's of a different blood type than our own. And this is a good way to try to avoid this type 2 hypersensitivity through the process of blood donation. Unfortunately, there's a natural situation where type 2 cytotoxic hypersensitivities arise in patients without the donation of blood, and that's illustrated here in this picture. A type 2 hypersensitivity happens with a mother bringing the baby to term when the baby is of a different blood type than the mother is. This is a phenomenon known as RH, Antigen Incompatibility, and during the first pregnancy, if the mother is RH negative and the baby is RH positive, actually nothing happens during this first pregnancy. The baby comes to term, and during the process of labor and delivery, there is some small amount of mixing of blood from the baby with the RH plus antigen, which gets out into the mom. This is where the trouble actually starts, because once this RH positive blood enters the mom's circulation, what ends up happening is that the mom ends up making an antibody against the RH antigen. This becomes a problem during the subsequent pregnancy, during the second pregnancy. During the second pregnancy, this RH antigen can enter into the baby's circulation because it's an IgG. The IgG can cross the placenta, and then in the second pregnancy, over here on the right, the IgG anti-RH antibody enters into the baby's circulation, leading to complement-mediated lysis of red blood cells, just like we saw in the previous slide. The resulting fetal red blood cells being destroyed can lead to all sorts of developmental issues as this baby comes to term. So, what can be done to avoid this? This situation, this RH incompatibility, only arises when the mom is RH negative, that is, the mom's blood cells do not have the Rh antigen on them, but the father's red blood cells do have the Rh antigen. Through the power of genetics, what this means is that the baby has a 50-50 shot, potentially, of having red blood cells with this Rh antigen. It's not a problem at all for the first baby because the mother has not had the opportunity to make antibody that reacts against the Rh antigen. It's only a problem with the second and third and subsequent pregnancies if the second or third baby is also Rh positive. What's done to prevent the development of these Rh positive antibodies is that all the way back over there at the left, with that very first Rh positive fetus, at the time of delivery, the mom is given a drug called RhoGAM, which you may have heard of. What is RhoGAM? RhoGAM is an antibody preparation that reacts against red blood cells with the Rh antigen. The red blood cells are actually coming from the baby going into the mom's circulation. By adding these Rh positive antibodies in RhoGAM, complement-mediated lysis occurs in the mom after delivery, but before the mom has the opportunity to make Rh positive antibodies herself. So this is a form of passive immunotherapy. RhoGAM is passive immunity that's giving to the mom. It's obviously artificial because we're giving an injection into the mom, but this passive antibody transfer given to the mom at the process of delivery of an Rh positive baby prevents the formation of anti-Rh positive antibodies and any subsequent type 2 hypersensitivities. All right. Type 3 hypersensitivity. These are known as immune complex mediated hypersensitivities, and again, it involves an antibody, generally IgG, although sometimes it can be IgM, an antigen which enters into the body, and the formation of antigen and antibody complexes, immune complexes. This is through the process of agglutination, which we recall is one of the properties that antibody molecules can have. As agglutination occurs, clumping of antibody and antigen, these can get trapped in various organ tissues, which are very good at filtering things out. These are primarily going to be tissues like the spleen, going to be like the liver, going to be like the kidneys. Kidneys are very good targets for immune complex deposition. As these immune complexes get stuck in places where the blood doesn't move through the circulatory system quite as quickly and easily, this can lead to complement coming in and initiating the process of inflammation. So we can see this sort of outlined here, the series of events going on in here. We have antibody of the IgG, primarily isotype. We have the presence of antigen forming immune complexes, and these immune complexes get stuck in various places, like in the kidney. Complement gets turned on, leading to the induction of inflammation. So inflammation is the ultimate result of this. When do type 3 immune complex hypersensitivities occur? Well, they typically are present when we have an inappropriate immune response against, for instance, one of our own antigens, an autoantigen. And so type 3 immune complex hypersensitivities are generally the basis behind a bunch of diseases known collectively as autoimmune diseases, where we have immune complexes, chronic inflammation, all directed against antigens that come from our own cells. Now, because the antigens come from our own cells, not from a pathogen, what this means is that ultimately that antigen will never be removed from the immune system, never removed from the body, and inflammation that gets turned on as we go through to step 5 is never going to get turned off because the initial trigger is an antigen found on our own cells. This leads to a chronic inflammatory situation. One example of a chronic inflammatory situation is the disease lupus. Lupus is a chronic inflammatory situation that occurs. It's a type 3 immune complex mediated autoimmune disease, the presence of antigens, autoantigens, and antibodies which have been raised against those autoantigens tend to get stuck in a number of places throughout the body. The capillary beds of the extremities have poor circulation, and immune complexes get stuck there. The joints have poor circulation going through them, and consequently immune complexes get stuck in there. The kidneys work wonderfully to filter things out, as do the hearts, as well as the lungs. Immune complexes, inflammatory responses getting on in there. Ultimately, the antigens can never be removed from the system. And so for a disease like lupus, there's really not any cure. Mostly what can be done with these patients is to give them supportive therapy. Anti-inflammatory drugs can begin to diminish the degree of inflammation that's occurring here, but ultimately it's just not feasible to be able to try to remove the presence of the antigen from their bodies. All right, and then finally, type 4 hypersensitivity, also known as a delayed type hypersensitivity. It also involves an antigen, but the antigen in this situation is very different from the antigen in the first 3 type hypersensitivities. In the type 4 hypersensitivity, the antigen is an endogenous antigen as opposed to an exogenous antigen. And as we recall from Chapter 16, when we talked about antigens, endogenous antigens are going to be found for the most part on our own cells, but they're going to be foreign antigens which are present on our own cells. And we remember this in the context of virally infected cells. Well, what's the other player with endogenous antigen? It's not an antibody. It's a T cytotoxic cell, a cytotoxic T cell. This T cytotoxic cell recognizes endogenous antigens, but instead of triggering something like agglutination or complement-mediated lysis, what occurs is that the T cytotoxic cell attacks and destroys directly the cells bearing an endogenous antigen through the process of cellular lysis. This is illustrated very nicely over here on the right-hand panel, where we transfer some tissue from one animal onto another animal. If the host and if the donor and the recipient are genetically extremely similar, the antigens are not going to be significantly different between the donor and the recipient. If we have a donor and a recipient which are genetically diverse, as in this white mouse and this kind of grayish mouse, they're the same species. Many of their antigens are going to end up being very, very similar to one another, but there will also be some significantly different antigens. In the case of this second situation, there's a good chance that the antigens that are on this donated tissue are going to be recognized by the immune system. T cytotoxic cells will see those endogenous antigens and lead to destruction of the donated tissue. This is the principle behind organ transplant failure. Now, when we have two members in a pair, a donor and a recipient, that are different species, as in the case of this mouse and this cat, the antigens that are present on this donated tissue are going to be extremely different. And the tissue that's been donated from the mouse to the cat is going to have a very high probability of being rejected extremely quickly, as the endogenous antigens on that mouse tissue are recognized by T cytotoxic cells and destroyed. From a clinical standpoint, this is the principle behind organ rejection, and any transplant patient has to face the possibility. In a donor and a recipient for a donated organ that are genetically extremely similar, the antigens will be highly conserved between the donor and the recipient, and consequently that donated organ is going to have a pretty reasonable probability of being successful. Most humans are not genetically very similar to one another, and consequently there are going to be pretty significant antigen differences between donors and recipients, and consequently the rejection of these donated tissues means that the recipient would need to be placed on immunosuppressive drugs for the duration of their life to ensure that they do not produce T cytotoxic cells recognizing the antigens on the donated tissue, and also to ensure that they don't reject this tissue. Well, hopefully none of us will need to deal with the possibility of an organ transplant, but all of us going into healthcare are going to have to deal with the situation that's over here in the left-hand picture. What we see in the left-hand picture is a cellular lysis phenomenon that is due to the presence of an antigen which has been stuck under the skin. The diagnostic test that's over here on the left is actually a test for the disease tuberculosis. In this situation on the left, the patient has been injected with a very small amount of antigen from the acid-fast bacteria, Mycobacterium tuberculosis. The antigens that are found on Mycobacterium tuberculosis actually associate with the cells under the skin, creating an endogenous antigen situation. Cytotoxic T cells, which recognize that antigen, will then slowly migrate under the skin through the tissues till they reach the site where the antigens are and begin to destroy the antigen-covered cells. These type 4 hypersensitivities are extremely slow in comparison to the first 3 hypersensitivities. This is because the antigen binding to T cytotoxic cells occurs much more slowly than the binding of antibody and antigen that we had in the first 3. Yet, in this case as well, this type 4 delayed type hypersensitivity, the positive reaction that we see in this TB test only occurs if the patient is producing T cytotoxic cells. Those cells are only produced if the patient has previously, before this TB test, become sensitized or exposed to tuberculosis antigens. And so the TB test here, this type 4 delayed type hypersensitivity, is actually used as a diagnostic to identify whether or not a patient has come into contact with the antigen and presumably the bacterium before in their immune history. Once a patient becomes positive with this, they can be further tested to see if they are in fact ill with tuberculosis. If they are, that can be, of course, treated with antibiotics, but they will never become unsensitized to this antigen. At any point in their lives after this, they will come up positive with this TB test. So from the point of view of somebody who is in healthcare, and we want to ensure that they are not carrying, they're not asymptomatic for TB, we can perform this test. If they come up positive, they will always be positive for this test, even if they are not asymptomatic, even if they are not carrying Mycobacterium tuberculosis. For those people who are unfortunate enough to come up positive with this TB test, what that means is that if they want to be in healthcare, if they want to be able to have patient contact, they will need to have an annual chest x-ray to assure that they are not carrying the microorganism in a carrier state.